for the first part of the lab was about cell size. And the big picture about cell size, that the larger your cell is, the more constraints or pressure, not literal pressure, but I mean like emotional pressure that puts on the cell in order to meet the demands of the cell. So the larger the cell, the higher the demands it would put on the cell, and the harder it would be, especially if we're talking about getting materials in and out of the cell, the harder it would be. So a hypothesis would probably say something about, increasing cell size would decrease the rate of diffusion. Your independent variable would need to be cell size, and then your dependent variable would be the rate of diffusion. But here's where it gets a little bit tricky, because as you can see in this lab, there were the 3 agar cubes, and they're actually not that much bigger than each other. They're only 1 centimeter. What was it? Was it their length? They had a 1 centimeter length, a 2 centimeter length, and a 3 centimeter length. So they are basic. They're very, very similar in size. So because of that, when you get down to the calculation of your rate, you'll probably find that these numbers are very similar. But the kind of catch-22 about it is that if you look back up here at the pictures, even though the rate of the 1 centimeter cube seemed to be the slowest, it diffused completely out of the cube, because 0.5, that rate, means that it made it through, or that, what was our indicator? Phenolphthalein was able to, vinegar was able to interact with all the phenolphthalein, diffuse across the entirety of that cell, and get all the phenolphthalein to become colorless. Whereas even though it seems like the rate of diffusion is longer for these two because it traveled farther, less of the color, less of the phenolphthalein interacted with the vinegar because the size of the cell was larger. So I just wanted to say that because I don't want you guys to think like, oh, and that means the smaller the cell, the better the diffusion. No, not really. You have to kind of keep in mind these pictures with this rate. So even though the rate may seem slower, in that same amount of time, it was able to diffuse completely throughout the cell and interact with all the phenolphthalein in the cell, whereas in the other two it wasn't. And obviously we can see in the 2 centimeter cell, more of the vinegar was able to interact with the phenolphthalein, and in the 3 centimeter cell, was the least amount of vinegar is able to interact with the phenolphthalein. So that's the big takeaway. I just don't want you guys to get too caught up on these rates of diffusion and then start thinking that smaller cell sizes are better, because that is the opposite of what you're trying to conclude. The biggest conclusion is here: if your cell grows too large, we're talking about comparing this to an actual cell, your plasma membrane would not have a sufficient surface area to support the rate of diffusion. So basically, as a cell becomes larger, it becomes less efficient. This larger cell wasn't able to get material in and out of the cell as well as a smaller cell was. And another thing to notice is that as your surface area increases and as your volume increases, your surface area to volume will decrease. That's another big thing. All right, and then there really wasn't any problems with the part 2 with diffusion. The biggest thing here was remembering that glucose was a monosaccharide and starch is a polysaccharide. So in terms of sizes of molecules, glucose is a smaller molecule and starch is a much larger molecule. So glucose is going to be able to get in and out of our cell membrane, which in this case was being represented by a dialysis tubing, but the starch is too large to be able to get in and out of the membrane. So at the end of the experiment, you would have glucose on both sides of the beaker, so in the beaker and in the bag, but starch will just remain in the bag because it's too large to pass through our cell membrane, our dialysis tubing. Now, this was the big one, though, that I really want to talk to you guys about, because the table is wrong. And I don't think anyone caught it. I didn't catch it until it was too late. I didn't make this lab, Mrs. Borgelt did. And so I just kind of trusted the numbers, but I shouldn't have. I should have gone back and looked because the numbers that you guys got for your final mass, I don't know what those numbers were. I think they're just like placeholders that she put in, but she forgot to change. If you actually watched the video, like part 2, you'll notice that those numbers are completely different from the actual final mass numbers that she got. and she says them out loud. So I would change these numbers to reflect the actual numbers that she says in the lab. And if you want to, because I have them up, just like take a picture of it, because I did the math here for you. So the benefit of you being here is you can just take a little screenshot. But it makes the results completely different, because if you try to graph percent change in mass based off these whatever numbers, your graph would have looked super funky like this. It just didn't make any sense. But if you use the actual final mass that was recorded and get the mass difference from that, and the actual, here I'm labeling it, actual percent change, your graph is going to look a whole lot differently. It's going to be a gradual increase. So we have here on the x-axis the different concentration or molarities, and then here on the y is the percent change in mass percentages. And so now you can see that as I increase the concentration or the molarity of my sucrose, my rate of, or my change in mass percentage is going to increase. And that makes way more sense. So this is really important, because if you use these numbers, your mass differences, your percent changes are all wonky. Your graph is going to be all wonky. And then your questions that you have here are going to be all wonky as well. So you need to make sure that you're using the actual numbers to calculate this graph to get the valid results. So yeah, that's it. I'm going to stop recording now in case, so I can post this one.